Hello everyone and welcome to this week's SEDS Online webinar. We're really pleased to be able to offer these events to you for free, thanks to sponsorship from the International Association of Sedimentologists. This week we are really pleased to introduce our first floating webinar. So from Mardi McNeil. Mardi completed her Bachelor of Applied Science in Environmental Science at Queen's University of Technology and then went on to complete her Bachelor's of Science in Earth Science with honours. She has received a number of academic scholarships and awards, including the 2018 Terry Walker Award from the Australian Reef Society. Mardi is presently a sessional academic and PhD candidate at Queensland University of Toronto in the Science and Engineering faculty. Her interest is in cross-disciplinary research in marine geoscience, specifically in biogenic carbonate sediments and structures at different spatial scales. So today she's going to be talking to us about building big bioherms from the humble Halimedas. So without further ado, I'll hand over to you, Mardi.
Thank you very much, Catherine, and hello everybody on shore, wherever you are joining us from. Thank you to SEDS Online for inviting me to give this talk today. As Catherine said, this is a floating seminar. I am currently at sea at the moment. So hence the portholes behind me, and if it looks as though I'm rolling around a little bit, that's because I am, but hopefully that won't be too distracting. Yeah, so today I'm going to be talking about building big bioherms from humble Halimeda. And if you're curious as to why I have a big green donut on the first slide, we'll get to that in a moment. So yes, I'm currently or near the end of my PhD studies at the moment, where I've been working on Halimeda bioherms from the northern Great Barrier Reef. So today I'll just give a few little background basics for those who are not carbonate sedimentologists and not so familiar with Halimeda. We'll have a look at some examples of some modern depositional environments, Halimeda in the geological record, and then we'll head to the GBR, Great Barrier Reef, and have a look at some of the work that I've been doing for my PhD. And that will include looking at the where, when, why, and who of the Halimeda bioherms in the Great Barrier Reef. And then we'll get to the donuts. Okay, so Halimeda is a genus of calcifying green macroalgae. It is what we call a bioconstructor. So it's a fleshy green macroalgae on the outside, and in the inside it has a calcium carbonate skeleton, which is aragonite. That's the same carbonate mineral that corals build their skeletons from. So we lovingly refer to these as limestone cornflakes. So Halimeda has a very high metabolism, and it's constantly growing and shedding these limestone cornflakes and producing sediment. So Halimeda is one of the most prolific sediment producers in tropical reef settings. It's estimated that a kilogram per square metre of live algal biomass can produce 2 kilograms per year of carbonate sediment, and that equates to a vertical aggregation or accretion rate of about 2 metres per 1,000 years. So a bioherm is an in situ carbonate biogenic mound-like structure with positive relief that is distinct from the surrounding seafloor or facies. So just an illustration here of a bioherm, which is mounded, as opposed to a biostrome, which is sort of flat and sheet-like. The key things are carbonate, biogenic, and positive relief. This is an example. This is actually modelled on the situation in the Great Barrier Reef, where you've got the reef on the edge of the continental shelf here and bioherms and biostromes behind. Here in the Great Barrier Reef, it's about 1 to 2 kilometres off of the back reef. And so in this case, Halimeda is our bioconstructor. So not all bioherms are necessarily constructed by Halimeda. You can have bioherms constructed by any carbonate constructor, so that might be brachiopods or can be anything, in our case Halimeda, and not all Halimeda builds bioherms. So a differentiation there is lithophytic versus samophytic Halimeda. So lithophytic Halimeda likes to attach itself to hard substrates, so rocks, rocky reefs, steep slopes, where it clings on by its sort of thread-like at the base. And samophytic means sand dwelling. So the samophytic Halimeda is the stuff that I deal with. It lives in the soft sediment and it produces the soft sediment, that carbonate limestone cornflakes. So some of the modern depositional environments where you will find Halimeda, because Halimeda is everywhere. So if you've got a core or a sample that's full of Halimeda, it doesn't necessarily mean that it's come from a bioherm. So Halimeda is really prolific on shallow coral reefs, so up in amongst the coral, within sea level to 5 meters. And then you can have these meadows, and so the meadow is the living veneer of Halimeda, which can be sitting on top of a bioherm, mound, bank. They're kind of just different terminology for more or less the same thing. So a living meadow on top, and the bioherm is the limestone cornflake sediment that's built up underneath. And then, so this scale is meters below sea level. So then at mesophotic depths, you can have mesophotic meadows and draperies that is more the lithophytic, so it attaches to the hard slope there. And then at the base on the seafloor, you can have debris from all of the above. Here's an example of a modern rudstone that I collected recently, actually from the ship that I'm on now, about a month or so ago. So this has just been brought back to the lab, and I haven't really looked at it yet other than take photos. Collected from about 80 meters of water depth. So it came from a mesophotic meadow. And here's an example of a fossil rudstone that I collected, and it was from 589 meters deep. Water depth is where I collected that from. So how did I do that? I used an ROV, a remotely operated vehicle with manipulators that can just reach in and grab samples. That makes it sound very easy. It's not quite that easy. And just to zoom in of that fossil rudstone there. So the scale here is centimeters, and you can see that that's packed with Halimeda flakes that are now cemented together. So Halimeter in the geological record. This is just a map showing some global locations that have been described in the literature, in their respective papers, as being bioherms or banks. So of course there's Halimeter in lots of other places, but these are the pins where it's been described as a bioherm. So as you can see, most of the Holocene and Pleistocene is around sort of the present day Indo-Pacific. And then there's some fossil outcrops now that are obviously on land, and you can go look at those in outcrop. So this is an example of a seismic profile from the Great Barrier Reef. So this is the Holocene build-up of limestone cornflakes, and it sits above the Pleistocene erosional surface. So the top of the bioherm is in about 30 meters of water depth, and the thickness of sediment build up there is about 20 meters over the past sort of about 10,000 years of the Holocene. The scale is seismic two-way travel time, and the scale is meters. And another seismic profile, this one is from K-Bank in Indonesia, and you can see that same or very similar sort of sinuous bioherm mounded build up there, sitting above Pleistocene erosional surface. So here's an example in outcrop from the Sorbas Basin in Spain, from the Upper Miocene. I have never been here. In fact, I've never seen any Halimeda in outcrop, I don't think. So if anyone is running a field trip to the Sorbas Basin and would like to invite me along and sponsor me to go, feel free to get in touch. So a lot of similarities here to the Great Barrier Reef, where you've got this mounded build-up that's about 25 meters thick. And bear in mind that there'll be a lot of erosion that has already taken place here. So these build-ups must have been really substantial in their day. The Halimeda is sitting above a Parietes reef, a coral reef, and I guess this is an erosional surface in between. So very similar, again, the term that we all use, an analogue between the Great Barrier Reef and what we're seeing in the Sorbus Basin in Spain. Going further back in time, the Halimeda bioherms, modern and Holocene, particularly the Great Barrier Reef, have of course been compared to the phyllowid algal mounds from the late Paleozoic. So just an SEM image here of modern Halimeda and Ivanovia from the Permian and Eugonophyllum from the Carboniferous. These are thought to be green algae and aragonetic. And here's an example of an outcrop of a phyllowid algal mound in the USA. And you can see that similar sinuous mounded sort of peaks and troughs morphology that's quite similar to how the Halimeda expresses itself in the Great Barrier Reef. So you can see why people have made this comparison, although it's not thought that they're taxonomically related, but there's certainly the mineralogy is the same, and it appears that the geomorphology is very similar as well, and probably occupied a similar shelf edge or platform top position as well. So this is something I put together through my PhD. So I've been working on 10, or more than 10 actually, probably 13 different Vibro cores that were collected in the Great Barrier Reef. They weren't collected by me, but I've found them in storage and have been working on those. So each Vibro core is only about 5 meters long at maximum. So I've put together this sort of composite core where I've kind of mashed together the different cores that were laterally related and to get these transitions. So what I'm dealing with is the Holocene section, post the last end of the last ice age, where post-glacial flooding, the Great Barrier Reef was once exposed on land and that's been flooded. And then the Halimeda carbonate factory has kicked off quite soon after the marine transgression. Just some images of these different lithologies. This is what the Halimeda looks like right at the top of the core. You can see that it's still articulated. So each Halimeda segment is still joined together at the nodes between each segment. And as you go down core, it's a bit more fragmented and lots of forams in there as well. At the base of the core, prior to the Halimeda having kicked off, it's very rich in small benthic forams. And then below that is this sort of shelly marine fauna that signals the onset of marine conditions. And I was lucky enough to find paleosol and a mangrove peat at the base of some cores as well. So mangroves, of course, grow sort of in the estuarine intertidal zone, and then the marine conditions, and then deepening upwards as the shelf was flooded. So to the Great Barrier Reef. This is the ship that I'm on right now, fortuitously and coincidentally, not related to my PhD work. This is like a side project tangent that I was lucky enough to be invited to come along and be involved in. But this is me doing some of my field work in the Great Barrier Reef. So this is my version of walking the outcrop because I don't have any outcrop. So most of my Halimeda is too deep to dive on, but I did manage to find one location where it was 25 meters and that was shallow enough to allow me to scuba dive, because come hell or high water, I was going to dive on my Halimeda bioherms. So that's me collecting some sediment from down there. Generally, for most of my video footage, I've used a little small remotely operated vehicle, a little ROV, which is tethered to a laptop on the ship. So send that down and fly it around and have a look at what this stuff actually looks like down there. So this is northeastern Australia, Queensland, and the inset that I've circled here is just one section of the Northern Great Barrier Reef where there are Halimeda bioherms. And they lie in what we call the interreef lagoon. So this line of reefs here, these are the outer barrier coral reefs. So sometimes called the ribbon reefs. And sitting just landward in about 25 to 50 metres of water depth is where we find the Halimeda bioherms, not just in this discrete location, but from up here all the way down to about here. So that's the where. So here's some mapping that or the result of some mapping that we did a few years ago now, back in 2016. So the grey here are coral reefs, and these are the Holocene modern coral reefs that you can see at sea level now. And then sitting just behind that in the green is the distribution of the Halimeda bioherms. They span 6 degrees of latitude and cover over 6,000 square kilometres, which I think once upon a time I worked out that that's 110 Sydney harbours, if that is of relevance to anyone. In some places, the Halimeda bioherms occupy up to 25% of the area of the continental shelf. And this little 3 times the original estimate, that's from some single beam echo sounder work and some surface sediment grabs that were done in the 1980s. And it was estimated that these Halimeda meadows or banks covered about 2,000 square kilometres. But we've come along with high resolution bathymetry mapping and yeah, over 6,000 square kilometres. It's pretty substantial. So I think I'm up to the when. So as I mentioned, I'm kind of dealing just in the Holocene. And what you're looking at here is 12,000 years before present towards present day on left. And the y-axis is present mean sea level. And so this is a sea level curve for Eastern Australia. And so, as I mentioned in a few slides before, I was lucky enough to have some mangrove peat in the base of my cores. So this is when it was a very shallow intertidal estuarine salt marsh mangroves type environment. And as sea level has risen, we've got those marine molluscs, that shelly fauna coming in. And then very soon after, the Halimeda first appears in my cores. So the Halimeda kicks off and is depositing carbonate. And then that is quite some time before the Holocene coral reefs actually initiate. So down in this panel, I've just kind of put that more or less the same information, but presented in this way. So mangroves and then the forams kick off. Halimeter is going for more than or about 2,500 years before we see the first coral reefs initiate. And I've just put this together as a bit of a cartoon diagram. So pre-Holocene, what is now the Great Barrier Reef or Australia's continental shelf was high and dry. It was dry land, and it was a coastal plain and actually a tropical savanna. And I know that because I sent that mangrove peat sample away for pollen analysis. And so we came back with this sort of tropical savanna type vegetation community at that time. And then the marine transgression has started to flood the shelf, and we change our environment to an intertidal estuary. We've got lots and lots of mangrove pollen in the samples and some sort of salt marsh rushes and things like that. And then as sea level has continued to rise, then that was conditions that allowed the Halimeter to kick off. And then by 8,800 years before present, we get the first coral reefs start to initiate. By this stage, the Halimeter is well established, and these bioherms are just continuing to grow and accrete to the present day. So the why are they there? Why are they there and not in other places? And the hypothesis is that the nutrient source, it's related to the nutrient source. So it's a macroalgae. It likes lots of nutrients. which is a bit of a paradox because it's in a tropical reef environment, which is typically oligotrophic or very low in nutrients. So the hypothesis is that the nutrient source supplying the Halimeda bioherms originates from deep oceanic upwelling from off the continental shelf. And similar has been thought or assumed with respect to Halimeda bioherms through the fossil record and with the phyloid algal mounds as well, that they're kind of nourished by upwelling. And that idea, when you follow it back through the literature, it seems to have originated from the work in the Great Barrier Reef. There was an oceanography experiment that was done, and they followed the water mass, and the water mass from off the shelf seems to track up and intrude onto the shallow continental shelf. So we wanted to see whether we could actually test that geochemically and find some kind of geochemical signature for that upwelling. So this is what I'm talking about here. So here's sea level, and here's the Halimeda bioherm sitting in behind. And you've got a thermocline sitting kind of out here with mixed warm water above, but cold, nutrient-rich water below. And so this would be upwelling up the slope and intruding onto the shelf. So to test that hypothesis, we undertook some nitrogen isotope analysis. So a delta 15N from the Halimeda skeletal organic material. And so it's the nitrogen from the organic material that is actually locked within the aragonite crystal lattice. So we know that it got trapped in there at the time that the Halimeda was living and growing. And it's the ratio between 15N and 14N. And different parts of the ocean have a different delta 15N signature. We know from the literature, and this includes in the Great Barrier Reef, that this reef called Halimeda that sits up in the shallow in amongst the coral reef has a delta 15N signature of around 2 to 2.5 per mil. And we know that in the Southwest Pacific Ocean, where we are in the Coral Sea, the delta 15N is typically between 5.5 to 8 per mil. So my hypothesis was that the bioherm Halimeda, if it's being nourished from this upwelled water, should have a delta 15N signature within this range, and not the 2 to 2.5 that you get up in the coral reef. This is sort of the result, and there's lots of numbers and things going on in this slide, so I'll try and talk it through. So this is the Halimeda bioherms sitting in the lagoon in between the coral reefs and the passage there. This would be the conduit, the pathway for upwelling. And our Halimeda bioherm signature was 6.43, so well within that 5.5 to 8. And what we were able to match that with, this is a water column profile from the western subtropical Pacific Ocean. These abbreviations, sorry, I haven't included a key here, but these are just the different abbreviations for what we call these water bodies. So subtropical mode water, subantarctic mode water, it's not important, but this layer has a delta 15N signature of 6.3 and a nitrogen concentration of 7.4. And our nitrogen concentration was 7.9 micromolar, and the Halimeda was, as I said, 6.43. So I think that we have fingerprinted the water mass that is intruding up, and it confirms this hypothesis of oceanic upwelling, and is a very good match for this particular water body, which is all down our coastline and would intrude onto the GBR shelf during episodes of upwelling. So that also, where we get this upwelling does seem to correlate very well with where we find the present day distribution of the Halimeda bioherms. So this panel here is a diagram of the main oceanic currents. Here's Northeast Australia, Northeast Queensland. This is the Great Barrier Reef down the edge here. And we've got this south equatorial current that moves westward. and then it branches off and it heads to the north and to the south in the East Australian current, and it's bringing all that nutrient-rich water that then hits this barrier, the boundary current, and then intrudes up onto the Great Barrier Reef. And this little inset box here matches these panels here. Not sure how well you can see that, but there's a white outline here, which is the distribution of the Halimeda bioherms. And this scale is temperature in degrees Celsius. So obviously red for warmer, blue for colder. You can see that there's this cold water intruding on the particular day of that this modelling was run. This panel is nitrate, so red is more nitrate. So between the nitrate and the temperature, which is our signals for upwelling, you can see that it matches just pretty much perfectly with where the distribution of the Halimeda bioherms are. It appears that there are no bioherms in this bit here and there, and it also appears that upwelling doesn't intrude onto the shelf at that location. We're not quite sure why. We've got a bit more work to do on that. Yeah, this panel, so the bottom panel is my Halimeda nitrogen isotope results. We went down core back in time as far as 5,000 years before present towards today, modern day on the left, and the y-axis is our delta 15N. And we've got this pattern here, which also seems to correlate and match other patterns of climate variability. So in our part of the world, we talk a lot about ENSO, or the El Nino Southern Oscillation, and El Nino La Nina, and we're also kind of impacted by the Indonesian Australian summer monsoon. This panel is a oxygen isotope record from a cave speleothem in Northern Australia. And these top two panels are referring to this ENSO or El Nino frequency of events and the amplitude of events through this time period. And where we get our 15N minima seems to match up quite well with a period in time when we had a sort of about almost 1,000 years of like peak El Nino conditions. So this is the first time that Halimeda nitrogen isotopes have been done and used in this way. And so in this particular case with the nitrogen isotopes, it does show that for this purpose, they're quite a good geochemical proxy. And that seems to have worked quite well, which is nice for my PhD. Thank you very much. Okay, so who is living there in terms of the community of plants and fishes and invertebrates? So I knew that there was this biodiversity data set in existence in the Great Barrier Reef, and it only covers the inter-reef seafloor biodiversity, not up in the shallow coral reefs. So we're just talking about the inter-reef. And it was a massive undertaking, a huge survey that was done over 3 years by different Australian federal agencies. And I queried that data set for the community, especially of invertebrates that are inhabiting the Halimeda bioherms versus the inter-reef seafloor outside of the Halimeda bioherms. And it was, I found it all amazing. Hosting a high amount of biodiversity. The species richness in the plants, the algae, is not just Halimeda. There is a lot of other green macroalgae in there as well. The most frequently occurring invertebrates were sponges, a lot of diversity of sponges. And the invertebrates were mostly mollusks, echinoderms, and there was quite a lot of bryozoans in there as well. This footage was taken from that little remotely operated vehicle that I showed you on a previous slide. And this was the first time I ever flew it. And when I got to the bottom, I nearly landed right on top of that poor little hermit crab, but he seemed to be okay. And you can see his mound here is just all Halimeda cornflakes. So lots of bioturbation and lots of sedimentation there. So I ran some statistics on the community just to see how the biodiversity compared, and yeah, some graphs which are not so exciting, but what it shows, this one is average species richness, and this is all vertebrates, plants and invertebrates combined. The red is the Halimeda bioherms, and the blue is the interreef area outside of the bioherm boundaries. And I broke it down by plants, so obviously most of that is algae. Much higher species richness than outside of the bioherms. The vertebrates, species richness was quite similar, but the invertebrates again was significantly higher on the Halimeda bioherms versus outside. That's at the species level. And then I looked at the community level, the structure of the community. So not the number of species, but what species are present. So this is just a two-dimensional MDS plot at the community level. Again, the bioherm community is in red and the non-bioherm community is in blue. And they plot separately, which indicates that the structure of the communities are quite different as well. So Halimeda bioherms are highly biodiverse compared to the rest of the interreef, and it's quite a distinct community there. Okay, right, to the donuts. Where do they come in? So this is an interpretation of the geomorphology of Halimeda bioherms. Originally, this model of morphology came from like 1983, and it was derived from just two parallel lines of single beam echo sounder, and then basically they were interpolated between, and they came up with this kind of parallel ridges and troughs morphology, and that's permeated the literature ever since then. And then we came along with our high resolution multi-beam mapping and bathymetric LiDAR, and on the exact same area, this is what that part of the seafloor actually looks like. So the bioherm geomorphology is not linear, it's not ridges and troughs, it's donuts. So this sort of circular geomorphology, sometimes a bit irregular and sometimes very regular circular shapes. So this came as a big surprise, and this is not in one isolated area. This is what it looks like down that entire almost 6 degrees of latitude. It expresses this geomorphology. Each donut, if you will, is around 250 to 300 meters across in diameter. So these are big features. And we determined that there was sort of 3 different morphology types, and we termed them reticulate, sort of after reticulate reef patterns, very similar to that. Annulate meaning circular, that's the donuts. And then undulate is it's flatter and it's a bit more like a biostrome, and that kind of, it joins and connects and grades into the reticulate and the annulate. So donuts is the annulate, and you can see why. And we noticed that there was quite a cross-shelf gradient. So this red down here is the coral reef. So that's on the outside barrier reef. This is open ocean, and landward is to the left or westward. And we noticed that this morphology kind of grades across the shelf. We've got some thoughts about what that might be, but the cores that we have, the vibrocoring that we have, is very sparse and kind of not really enough to really query that to solidify some hypotheses, but we do have some ideas. I'm taking you through a LiDAR fly-through now, just to demonstrate how vast the seafloor is that's covered by the Halometer. And again, this is just one tiny discrete area. The red are the coral reefs, and then this sort of yellow-green colour is where the Halimeda bioherms are sitting behind. This blue shade is at about 45, 50, down to 55 metres. Up on here, it's about 20 metres deep. I'm not going to let you go to the next slide now. maybe I'll just go, there we go. So I recently did a bit of sort of geomorphology modelling and an investigation, which has just been published in the depositional record. It's open access, everyone can go check it out and have a look there. So I'm not going to go into a huge amount here, but some of the interesting things that we looked at and found that were quite interesting and curious. So we looked, slope was obviously one of the parameters that we investigated, and aspect, because we thought that if the geomorphology was being controlled by hydrodynamic processes, we might see some preferential aspect or slope that was kind of indicating sediment transport by currents and things like that. So just a little subsample of the annulate, the donuts here, and the reticulate. You can see that the expression is quite different. And what was interesting is with the donuts and the annulate, they've got two slope gradients. So they've got an inner slope and an outer slope, and the bright white is higher gradient slope. And so here is a profile going across one donut, sort of north-south and east-west. So they're very circular and in kind of both directions. And the slope, the inner slope is steeper than the outer slope. And we found that consistently. And then with respect to the aspect, you can see that it's a random jumble, and that's because it's not showing any preferred orientation or any preferred aspect in either the annulate or the reticulate. So their sides and slopes are facing in all directions equally. I don't know why this inner slope is steeper and the outer slope is a less steep gradient. If anyone has thoughts and ideas about that, I'd love to talk about it. Of course, we know about having a stoth and a lee, but this is circular, so it's curious. One of the other things that we've definitely found is that the surface morphology expression is not inherited from the antecedent topography. So this is a sub-bottom profile, and this is what we call reflector A. It's the erosional boundary between the Holocene and Pleistocene. There's all the bioherm build-up, and the erosional surface underneath is fairly planar. It has a gentle slope because we're on the continental shelf, but where we've got high mounds of Halimeda, it just doesn't correspond with having high topography beneath that. So the antecedent topography is not controlling the surface geomorphology. We found that there was no sort of preferred aspect orientation for any of the morphotypes. The reticulate generally has steeper slopes, like sharper crests and steeper slopes. And then the donuts, the annulate morphotype, has that differential slope gradient between the inner and the outer faces. The annulate morphotype has the higher sedimentation rate. There's thicker sediment packages over the same time period. So therefore, the annulate morphotype contributes significantly more carbonate deposition by volume. So what we do think is that the morphology appears to be modulated by biotic self-organisation feedbacks, which I'm not going to talk about too much because that could be a whole talk in itself. But it appears that the donuts, the crests of the donuts, are preferentially receiving that nitrogen nutrient input, and hence they grow a bit more. That causes a feedback where the hollow in the middle is lacking, and so that just you get more sedimentation, and once that pattern is established, it just kind of feedback on itself. That's a hypothesis. Again, I don't have enough cause to really test a few things around that. So our next question was, are there donuts in other locations? So there's Halimeda bioherms elsewhere in the Holocene and Pleistocene that are currently, they're subaqueous still now. And maybe what about in the geological past? So the answer is probably, we don't know. Here's a paper to go check out, which is from the fossil record that shows some reticulate patterns that have been interpreted as being from these self-organisation feedbacks. So the thing to bear in mind is that what looks like this in 2 dimensions looks like this in 3 dimensions. So to see this, you need high resolution bathymetry. So in our case, we've got LiDAR or very high resolution multi-beam bathymetry to see this. So what's next? Well, more cores. So we've been granted ship time on Australia's the Investigator. We were supposed to go in May this year, but of course it got cancelled because of COVID. And we've been rescheduled to July 2022. So we will be going back out and collecting more cores and doing more geophysics to try and unlock some of those other remaining questions around the processes around that geomorphology and lots of other things as well. So that is it pretty much from me. Of course, I work with a whole team of people, including my PhD supervisors, Luke Notiff and JD Webster, and a whole bunch of other collaborators that have helped me with things like the nitrogen isotopes and things like that. So thanks to a whole bunch of people. And that's it from me.
Thank you so much, Marty. Absolutely fascinating stuff. Always seeing imagery that you would never otherwise be able to see as a vision into a whole new world. So thank you very much. And it comes with cornflakes and donuts. So
yeah, the food analogy just keeps on giving.
We seem to have lost Catherine for a second, so while Catherine reestablishes her connection, I'll step in. As being as I'd actually written the first question. Oh, in fact, there's a question from Dan, but I'll read that one out, and then Catherine can read mine out. There you go. This is from Dan. He's watching live on his mobile from the Alps from Austria, anyway. Thanks for a wonderful talk.
Hello, Dan.
He says, thanks for a wonderful talk. My question is, what is the relationship between coral bleaching and Halimeda colonization? What do you think will happen over longer timeframes?
Sure. All right, so coral bleaching is a process that doesn't directly impact or relate to Halimeda in itself, but of course it's related to water temperature, sea surface temperature. So when it gets too hot, the corals, symbiotic algae, get stressed out and leaves the coral host. And then that's when you get bleaching. That doesn't happen in Halimeda per se because Halimeda doesn't host a symbiont. But it appears as though episodes of coral bleaching in the Great Barrier Reef appear to relate to hot El Nino summers, which for Halimeda, that then corresponds to our upwelling. So that's when we get most of our stronger upwelling. So it could be bad for coral, could be good for Halimeda. Maybe we're gonna, maybe on a geological timescale, we might get a bit of a phase shift where Halimeda algae becomes more prolific and more dominant, but that's purely speculation. I hope that answers the question. The second part, or was that it?
I think so. I mean, it's all guesswork when we move into the future, isn't it? Okay, right. We're still waiting on Catherine to come back, so I'll carry on until Catherine reappears. Hopefully she'll be able to re-establish her connection. I'm watching for her. Cedric, John, everybody send them to me privately. People, please, can you send them to everyone? Send them to everyone so everybody can see them, but I will continue reading out what I see here. Yannick Santera, I'm not sure where they're from. Yannick's asking, what is the largest bioherm height above the seafloor, please?
The maximum that I measured from seismic profiles, maximum thickness was about 25 meters thick, and the minimum was just 3 meters in the donut hollows.
Okay, terrific. Can I actually follow up on that? So the relief that you see of 25 meters, that's above the sea floor. Do those do they carry on constratally, substratally into the sea floor?
Yes, they do.
Okay.
Yes, you've got that Pleistocene erosional unconformity, and then some Halimeda, and then you'll have a donut hollow. So there's the seafloor in the donut hollow, and then on the crest it's still meters above that. So although your seafloor in the hollow is here, it's still, you know, meters of Halimeda there is below that as well.
Okay, terrific, terrific. By the way, anytime you can get to Sorbus, happy to show you around.
Yes, please.
Right, okay. I'm just sorry, I'm having to scroll down here because they're coming in so thick and fast. Cedric John from London, from Imperial. Great talk. Do you think the Halimeda bioherm shape is controlled by self-organizing feedback suggested by other carbonate fascies mosaics by Sam Perkis in Qatar, amongst others? If yes, what would control on these, and something in the ecological limitation of bioherm growth, maybe?
Yeah, so the short answer is yes. Hi, Cedric. I do. Our hypothesis is at the moment is that the most likely mechanism is probably this biotic self-organization as seen in other systems. And I guess potentially is two things. The higher topography preferentially being able to access the nutrient-rich water first, and then depleting that nutrient out of the water, and so the hollows miss out. And also potentially the living veneer of Halimeda meadow, sort of this carpet of living Halimeda, and then the bigger your meadow gets, the inside not potentially getting so much nutrient as the outside edges, but also perhaps some toxicity towards the center or lack of oxygen, causing a dieback in the center. And so now you've got a donut that's flat, and then add sedimentation into that, and you start to build a donut. So that's kind of our ideas at the moment.
Okay, terrific. I'll ask the next question because Catherine's just rejoining us at the moment. She's just coming into the room. While she does that, I'll ask the next question. This is from Elias Samancoso, who's over in Geneva. Elias is asking, thank you, Mardi. Great talk, impressive new findings. Do you plan to determine Halimeda at the species level to eventually evaluate the spatial distribution of various species?
Would you like to help me with that? So at the moment, I guess we talk about Halimeda just as a group, which is a genus. And of course, in the Great Barrier Reef, we have 14 different species, some which are the lithophytic, some are the samophytic, and different species in different sub-environments and what have you. I've got to be honest, I'm a little bit shy at differentiating at the species level, but what I would love to put together is an atlas or an identification key that works for biologists and ecologists and sedimentologists, where you've got the imagery of the whole plant, and then the individual segments, and then an SEM image of the skeleton, and a thin section under light microscope by species. That's not something I'm planning to work on anytime soon, but that would be what I would love to see. It would help sedimentologists, I think, immensely. And to look at looking at that species distribution, where it is of interest in these morphotype differentiation between reticulate and annulate and the undulate. Is it a species differentiation? So at the moment, we don't know. Thanks, good question.
Great, thank you. I'm going to hand you back to Catherine because she's back in the room.
Okay.
Hi folks. Yes, I'm not even on a ship and my internet went out. So thank you for bearing with. I'm just trying to, because I only have the chat from where we started. So is it which is the next question, Stephen?
Okay, sorry. The next question is from Dan. If you don't see that, I can carry on until. Give me the thumbs up when you can take over. Okay. So, right, Dan Mobile. Dan again from Austria again. This was my question to Mardi that she. Oh no, she's already sent it again. Okay. Or Bilik. From Israel. Do you have any samples or ROV from the center of the donuts?
Yes. my little smaller ROV, I put down when I knew that it was 25 meters water depth. So I knew that that had to be on the top of a donut. And then a little further along, the water was 45 meters. And so I knew that had to be in the donut hollow or off on the edge. So I put the camera down there as well. So I do have some images and footage from both. And there is some green living Halimeda down in the hollow, not much, but there is still some green Halimeda there. And that's 45 meters depth in the Great Barrier Reef, but I've seen Halimeda live and living and growing, and so have others, down to 135, 140 meters, obviously by ROV. No one's, well, some technical divers might go there, but I'm certainly not. So as long as you're in the photic zone, as long as there's sunlight, chances are there'll be Halimeda there.
Terrific. Yeah, going down on nitrox is not a good idea. Not unless you're very technical, as you say. Ezra from Turkey is asking, and this will be the last question from me, then back to Catherine. Thank you very much for the wonderful talk. I am wondering, what do you have, I'm wondering, do you have any idea for future climate change scenarios on their effects on the size of Halimeda?
So there have been a few papers come out now that have done, it's sort of been laboratory experimental stuff where they've manipulated temperature and they've manipulated acidity. And it's a little bit unclear because the Halimedas seems to do okay with increased temperature. There are some evidence at the skeletal level that acidification impacts the skeletal microstructure at that scale. So I guess given enough time, that would cause breakdown and erosion. So your bioherms, the Halimeter itself could become less calcified and more fleshy, which would then impact sedimentation. And then I guess at an extreme case, you could start getting, the bioherms themselves kind of eroding and flattening. Yeah, but that's all kind of laboratory stuff at the moment. I'm not sure in the real environment how that might play out.
Great. Our next question is from Yannick Santer, who says, are the whole bioherm tops alive or only a part of them?
So in the Great Barrier Reef, the carpet, the meadow, is all over the bioherm tops. But it is patchy in places. So yeah, you get patchiness like you do in seagrass meadows and forests and things like that. But essentially, yeah, the whole top is the living veneer, and then all that sedimentation is below that. So depending on, yeah, it's not wall-to-wall green Halimeda living 100% cover as far as the eye can see. The cover is not 100%, a little bit patchy, but you don't have to go too far to then see more.
Great. Next question is from Cedric, who says, thanks, Mardi. Follow-up question. How do your Australian bioherm compare to the classical Bahamas Halimeda meadows?
Well, I'm not sure if the Halimeda meadows in the Bahamas are forming donuts. Maybe you can tell me or follow up with me on that. But I guess they're a similar thing in that sense. Is that it looks like if you don't have geophysics and you're looking at the subsurface, then at the surface it just looks like a meadow. You wouldn't know that there's that thickness of sediment package underneath it if you only saw the surface expression. So a living green meadow.
Great. Next question is from Noah Subi. Hi, this is Noah from Hungary. Thank you for the nice presentation. My question is the climate change effect on extended of Halimeda?
So right now, I guess we don't have a long enough record observations from Halimeda in the Great Barrier Reef anyway to know, like, now in the short term. But in the long term, some of the impacts could be ocean acidification. rising temperature will be okay for a while, but then I guess it'll reach a threshold where too hot is just too hot. But then the other issue that could be impacted is ocean circulation. So changing circulation patterns might disrupt that upwelling, or it might enhance it. That's a bit unknown at the moment, but I think the short answer is that there will be change.
Great. Next question then is from Stephen from Wales. The images of articulated Halimeda in core are terrific. At the beginning, you showed a Halimeda rudstone collected from about 500 meters depth. What was the nature of the cement?
Lovely question. I might have to get back to you because I literally collected that sample on the last trip that I was on at sea, which was a month ago. And at the end of the voyage, I packed up all the samples and shipped them off to another lab. So I haven't made thin sections or anything yet. So just what I've seen in hand lens. So it looks like it's a isopachus rim of cement around each individual grain and then cemented between the grains as well. But I reckon as soon as I make a thin section of that, I will share that with you.
Great. And additionally, how do you see ocean acidification affecting Halimeda in these eutrophic settings?
So yeah, I guess in summary, if ocean acidification becomes quite enhanced, I think we'll see Halimeda having lower calcification and so becoming less calcified and more fleshy, potentially.
Well, we'll have to watch this space.
Yeah, I think watch this space. But I think there's a few years to go before we get to that point. So I'm looking forward to the rise of Halimeda. I wouldn't say I'm looking forward to the demise of coral reefs. I'm not saying that. But yeah, I think watch this space.
Fantastic. Well, I'm not exactly sure when I cut out before. So I'll just remind you that if you have further questions or you think of some later, or if you're watching the recording, please head over to the forums to find the dedicated thread and we can continue this discussion for as long as we need to. Thank you very much for your amazing questions from Israel, Wales, Turkey, Hungary, many places. And we hope to see you at next week's webinar, which is on the magnitude and drivers of short-term Cretaceous sea level fluctuations, a review from Franz Buchem and Andy Davies from Halliburton. So in the meantime, before we say goodbye, thank you again so much to Mardi, and thank you to everyone here for coming along and asking great questions. I'm sorry my internet dropped out, but you know, these things happen in the crazy COVID world. But thank you so much, Mardi, for coming along. And in the meantime, before next week, if you want some more SEDS online, come along to a coffee break. Thank you very much for coming along.
Thank you very much, everyone, for joining, and thanks for your questions.
No problem.